So we have recently applied this methodology to study one of the enzymes involved in GPI anchor biosynthesis pathway called TBGPI2. And we were able to identify the function of TBGPI2 and its involvement in the glycan biosynthesis. And after I purified the prostaglandin, I did the aquacitive treatment to get rid of the protein and lipid. And then I further permethylate the glycans, and then I analyze them with electrospray mass spec. And in the MS1, we get these beautifully triply charged precursor ions. And when we fragment them in the CID for MS2, we get this doubly charged ions after the rearrangement of the C2 position in the glucosamine. And then we further fragment in MS3, we get beautiful fragmentation pattern of the glycan. And based on the fragmentation, we know that what could be the structures in the knockout cells and what are the structures in the wild type cells. And compared to that, we were able to identify the function of TBGPI in the GPI glycan synthesis. And the study done on TBGPI2 is published now as well. So far we have seen that we can identify big glycans like N-glycans and GPI glycans through mass spec. But can we identify small precursors as well through mass spec? And the answer is yes, we can. So in case of GPI biosynthesis, we can identify pretty much all the precursors using mass spec. The only limitation sometimes comes is the quantity, because these genes are not expressed at a very high level, but it is not that difficult. For example, here we have recently characterized some GPI precursors of Leishmania donomani, and we were studying one protein called LGPBN1 with a putative activity of mannose-transferase. So the GPI anchor biosynthesis begins with a phosphatidylinositol, a phosphoinositol structure, and then a gluconic is transferred, which is further get deacylated, and then a mannose-transferase come, or MT1 it's called, which uses a dolly called phosphomannose as a donor and transfers a mannose on this glucosamine here. And the LDPBN1 again was a putative mannose-transferase, but we had to confirm the activity. So what we did here was to purify the glycogen. We extracted the GPI glycolipid and analyzed them electrospray in negative ion mode. And we could see that in MS1, PBN1 knockout had the accumulation of glucosamine BI precursor, which were not present in the add-back cell lines or wild-type cell lines. Because in the wild type and in the add-back cell lines, the precursors are being used for the GPI anchor biosynthesis pathway completion. But in the knockout cell lines, the accumulation of the precursor was suggesting that the PBN1 knockout lacks the mannose-transferase activity, and that's why leading to the accumulation of the precursor and not being utilized. And this analysis led us to help identify the activity of PBN1 in the Leishmania donovani. And the identity of these two species, which we identified to be glucosamine PI, was confirmed by MS2. And we identified these two species to be glucosamine PI, differing in the acyl chain at the SN1 position. So one is having C16 fatty acid chain, while the other one, the bigger one, is having the C18 acyl chain. And that's how we identified the precursors in the Leishmania biosynthesis pathway, GPI biosynthesis pathway. And this study is also would be published soon, as it is accepted already. Lastly, I would describe the method to analyze gippals, taking T. cruzi gippals as an example. I won't go into detail of the extraction process as this is a common extraction method which uses chloroform methanol water extraction and butanol water extraction. And this is a common way of extracting and fractionating glycoconjugates. So after we do the extraction and when we run these glycoconjugates on the gel and do a staining with pyridate shift staining, we can see how abundant these surface glycoconjugates are. As you can see here in the T-cruzi, we can see a lot of mucin proteins, which are again GPI anchored surface glycoconjugates of trypanosoma cruzi, and gippals on the bottom here, which are again heavily glycosylated and heavily abundant in the trypanosoma cruzi. And so when we run these purified Gippel fractions on electrospray mass spec, we can identify different Gippel species in Trypanosoma cruzi. And with the mass spec, we could identify at least three different types of Gippel species in Trypanosoma cruzi. As I mentioned earlier, that GPI anchor glycan core can be modified further with glycan residues or chemical modifications. And the two most common chemical modifications are either addition of 2-AEP, which is 2-amino-ethyl phosphonate, or ETNP, which is ethanolamine phosphate. And the three species that we identified in Trypanosoma gippals were differing in these chemical modifications, but having the similar lipid backbone. And that's how we were able to identify how diverse the Trypanosoma gippals can be. So we have seen that how mass spec-based approaches are so sensitive and provide us a great detail of these glycoconjugates. So to summarize my presentation, we have seen here that the glycome of the parasites is very diverse and complex, and the glycoconjugates are the major surface molecules of parasites, thus mediating the host pathogen interaction. Mass spec based approaches, be it JCMS based or ESMS based, provide a selective and useful approach to study these glycans and glycoconjugates. Therefore, glycomics and glycoproteomics based approaches are crucial in order to extend our knowledge about the biology of these pathogenic organisms and to be able to characterize the glycan based antigenic molecules. Finally, I would like to thank all my teammates and all my collaborators, also the fantastic proteomic facility that we have here at University of Dundee. I would also like to thank all the organizing committee members for organizing this event and giving me the opportunity to speak about my work. Lastly, I would like to thank everyone for listening. I'm happy to take questions if any. Thank you.